hope you can hear me all. Also, hello from my side. I'm Daniel. I'm a postdoc in the lab of John Kagan at Boston Children's Hospital and Harvard Medical School. I'd like to tell you a little bit about our efforts on defining the endogenous toll-like receptor signaling pathways in mammalian innate immune cells. And now I've been studying immunology for about 15 years now, but after attending this meeting for the last 3 days, I realized just how incredibly diverse this audience is in their scientific interests. So I'd like to start with a little bit of background just to get everyone on the same page, beginning with significance. Why should we care studying mammalian immunology? And for this, I'd like to remind everyone that we all live in a sea of microbes. We're surrounded by them. They're in the air we breathe, they're in the food we eat, they're in the water we drink. Yet we rarely fall ill, despite these microbes also containing some pathogens that when we encounter them, they can infect us, they can cause serious illness and even death. And one reason that we rarely fall ill is that evolution has equipped us with quite powerful immune systems that constantly work to maintain our health, and often we don't even notice this process happening. Now, we immunologists usually distinguish two parts of mammalian immune systems. We have the innate and the adaptive immune system, where the adaptive immune system is probably the best weapon in our arsenal. So these are T cells, B cells, antibodies, all of those highly efficient, highly pathogen-specific mechanisms that in a best-case scenario lead to a sterilizing reaction, restore homeostasis in our health, and also provide us with an immunological memory. But for this system to become activated, it can usually take a few days. And to bridge this gap in protection, we also have the innate immune system, which can respond within minutes after encountering a pathogen, is however considered non-antigen specific because it relies on broad antimicrobial mechanisms of action. And now, the other critical task that the innate immune system serves is the actual sensing of the presence of a pathogen. And that's probably the one central dogma of immunology, is that without a response by the innate immune system and forwarding this information and licensing the adaptive immune response, there would be no adaptive immune response. And so my work mostly revolves around this initial task, the initial sensing. And since this is such a critical process, our bodies have come up with several ways by which this can be achieved. The one type of mechanism that is most relevant to my work is the sensing of conserved molecular patterns. And depending on their origin, we distinguish between pathogen-associated or damage-associated molecular patterns, PAMs or DAMs. A classic example for a PAM would be LPS, or endotoxin from the outer membrane of a gram-negative bacterium, which is usually absent from a healthy tissue. An example for a DAM would be released cytosolic content, such as actin or ATP in the extracellular space. And so to sense these molecular patterns, our innate immune cells can rely on a class of so-called pattern recognition receptors, or PRRs. And these come in different flavors. We have toll-like receptors, C-type lectin-like receptors, NOD-like receptors, RIG-I-like receptors. And they either localize to the plasma membrane, the endosome, or the cytoplasm, and thereby really survey every single compartment in an animal's or a human's body. Now, the star of today's talk are going to be the toll-like receptors, which can be really considered prototypical pattern recognition receptors because they have been intensely studied for about 30, 40 years now, and they have taught us a lot, not just about their own biology, but about this entire concept of pattern recognition as a whole. Toll-like receptors are membrane-bound receptors. There's 10 members in humans, 12 functional members in mice. And about half of them localize to the plasma membrane, where they survey the extracellular space for the presence of cell wall or cell envelope components of microbes. And the other half localize to the endosome, where they respond to the presence of nucleic acids. And what is generally considered the mechanism of activation is dimerization upon binding their ligand. And this first dimerizes their intracellular domain, which in the case of toll-like receptors is called a tear domain. And we've heard all about those domains in a talk yesterday morning already. And what happens is when the toll-like receptor tear domain dimerizes, the toll-like receptors seed the formation of a signaling adaptor complex, or signalosome. Now, most toll-like receptors use a signalosome known as mydosome because it's assembled around the central scaffolding protein myddosomes, and it also includes kinases of the IRAC family, such as IRAC 1, 2, and 4, and the 3 ubiquitin ligase TRAF6. And this then, in turn, activates distinct downstream signaling pathways, such as the MAPKs, the kinase TBK1, and the IKK complex activating pro-inflammatory NF-kappa B signaling. And the concerted action of all of these signaling pathways then robustly reprograms the TLR-stimulated innate immune cell, leads to the secretion of pro-inflammatory cytokines, chemokines, type 1 interferons, initiating first a local inflammation and then a more systemic response. So this is how I drew the toll-like receptor signaling pathway last year. This is how it was drawn 25 years ago. And I hope you can appreciate that not much has changed since then, despite intense research. And there are several reasons for that. Most of these proteins are expressed at a really low level, which makes working with them difficult. And this is further complicated by the fact that we also don't have any good antibodies for most of them. So we're mostly just left with genetics. So we could create knockouts of these individual factors, see if they have a toll-like receptor signaling associated phenotype, and then roughly place them in this textbook schematic as we like to draw it. But that didn't actually tell us anything about the cell biology of how these factors work together. And so some of the open, unanswered questions that I wanted to address with my project were, where in a cell can we find these complexes? How long do they exist? How long do they signal for? How are they connected amongst each other? And most importantly, this is how we draw the signaling pathway in the textbook, but is that how it actually works? And so to overcome these technical limitations, we decided to take a different approach. And instead of creating knockouts, wanted to target this central scaffolding protein, MyD88, with a multifunctional tag using CRISPR knock-in technology. And the tag that we designed, I usually refer to as the AGF tag because it has three parts. It has the peroxidase APEX2, it has monomeric eGFP, and a triple flag tag, and that allows us to do proximity biotinylation and mapping the components of these complexes. We can do live cell imaging to map their dynamics and localization, and we can do easier biochemistry. And so we appended the sequence for this tag to the 3' end of the MyD88 open reading frame using a technique known as CRISPaint, thereby creating human and mouse macrophage cell lines with a C-terminally tagged protein, as you can see here on this Western blot. What you can also see on this Western blot is that MyD88 is actually a small protein, and adding this tag almost triples its size. So we got quite worried that we might just break the function of this protein and thereby toll-like receptor protein signaling altogether. And so we started by validating these cells. We checked MyD88 mRNA abundance, and there was no difference between the wild type and our AGF knock-in cells. We also did functional validation, looking at toll-like receptor responsive gene activation by qPCR, signaling pathways by immunoblotting, receptor endocytosis by flow cytometry, and the secretion of cytokines by ELISA. And in none of these assays could we detect any difference between the wild type or the AGF knock-in cells, neither in the kinetics nor in the magnitude of the response, which means these cells might actually be useful for studying toll-like receptor signaling. We then went on and tried to reproduce results that had been published before by other labs who showed that upon activating toll-like receptors, MyD88 assembles into small membrane localized clusters, which they thought were mydosomes. And indeed, when we use our macrophages, treat them with LPS, a toll-like receptor 4 agonist, MyD88 quickly assembles into these membrane localized clusters that you can see here appearing as these black dots. And we imaged this using turf microscopy to really limit ourselves to the processes at the plasma membrane. You can also quantify this. You can see that most complexes contain between 5 and 6 MyD88 protomers, and that they appear very quickly after activating signaling by adding LPS, but that they're only at the membrane for about 10 minutes, and after 30 minutes, they are completely disappeared. But that was actually a problem, because my data and previously published data would suggest that these complexes have a half-life of between 2 and maybe 5 minutes. But if we look at the biochemical definition of a mydosome, i.e. the interaction between MyD88 and the IREC kinases, then they should persist for an hour or even up to 4 hours. So to address this discrepancy, we next looked at their assembly with higher temporal resolution using co-immunoprecipitation assays. And as the turf imaging would suggest, we could see that indeed MyD88, after treating cells with LPS that activates TLR-4 signaling, the protein quickly interacts with the receptor. This interaction is highly transient and is lost. But if we look at the assembly of enzyme-rich signaling-competent mydosomes, we can see that this persists for much longer, and that this is actually when signaling is activated, as assessed here by the activation of a TLR-responsive MAP kinase that is p38 alpha. But this also means that mydosomes exist in a toll-like receptor-free state for the majority of their lifetime. And we could actually show that this interaction is stable for up to 12 or even 24 hours after treating cells with a toll-like receptor agonist. So where do these complexes go? And what we thought is happening is that they must dissociate from the receptor and to the cytosol. So we next looked by confocal imaging, and what we could see is that indeed MyD88 assembles into signaling clusters in the cytosol. We're rather surprised though, because they were reported to be static and receptor linked, but what we could see is that they're neither static, they're highly dynamic, and they also form dozens of clusters per cell that also, biologically speaking, are huge. Some of these can exceed diameters of 1 micrometer. And so we decided to change the nomenclature and refer to these small clusters at the membrane as protomydosomes, because what we think is happening that these protomydosomes dissociate from the receptor, enter the cytosol, and then act as building blocks for these larger clusters, which we now refer to as mydosomes. We can also quantify this data. We can see if we treat cells, the majority of cells quickly respond by assembling these clusters. We could show that the majority of newly formed mydosomes appear within the first 3 hours. That's when they're most dynamic, and they show a lot of merges, which leads to an increase in their average size. They then enter a sort of steady state phase where we have constant merge and split rates and stable numbers. And then after about 6 hours, they become more static. They start falling apart. Their number increases and their average size decreases. And after 12 hours, we're basically just left with inactive remnants. This doesn't just happen with LPS. This happens for all toll-like receptors, with the expected exception of toll-like receptor 3, which does not use MyD88 as a signaling adapter, and toll-like receptor 9 in human macrophages, as these cells simply don't express this receptor. And so to summarize this first part, mydosomes are not static receptor-linked signaling adapters, but instead they're seeded as protomydosomes, which are released into the cytosol, and they form a highly dynamic signaling compartment that can persist for many hours before they become more static, start falling apart, and then the remnants are eventually cleared by autophagy. What I've not shown you so far is if this actually matters for the signaling process. And so to address this, we next created a reporter cell line in which we express an M-plum-tagged allele of the NF-kappa B family transcription factor P65, and we can thereby follow its translocation into the nucleus and assess whether a cell has active NF-kappa B signaling or not. And when we combine this with our mydosome live imaging, we could show that cells first assemble mydosomes and then activate NF-kappa B. And when we analyzed this on the single cell level, we could also see that all cells that had mydosomes eventually activated NF-kappa B, and vice versa. All cells that had active NF-kappa B also contained mydosomes, which means that for us that these two processes are functionally linked. To see how mydosomes could mediate all of this, we next decided to map their composition over time using proximity biotinylation and highly multiplexed mass spectrometry. And after careful filtering and enrichment analysis, these are some of the hits that we got. So you can see mydosomes seem to always contain IREC kinases, IREC2 and 4, and this was a great internal control because that is the biochemical definition of a mydosome. You could also see that if we use a synthetic toll-like receptor 3 agonist, P3C, MyD88 at early time points also interacts with the receptor, but that this interaction is lost, which was highly reminiscent of TLR-4, and it was great to see because we don't have an antibody for this protein at all. The surprise that we got was that we can also find all of these signaling enzymes. We found the entire IKK complex. We found TBK1, and we even found the terminal effectors of the MAPKs and the NF-kappa B signaling cascade as part of these mydosomes, and they were thought to be downstream of it and not part of these clusters. So we confirmed this first by co-immunoprecipitation assays, but since these were both bulk assays, we next performed immunofluorescence imaging, staining the cells for all of the factors that we found by mass spectrometry, thereby creating a map of mydosome composition over time on the single cell, single complex resolution. And as you can see, again, mydosomes always contain IREC kinases, the E3 ubiquitin ligase TRAF6. They're highly ubiquitinated. They don't show any significant overlap with any other cellular compartments. And indeed, they seem to contain these signaling enzymes at early time points. The other thing we did here was use antibodies for their activated, i.e. phosphorylated species, which would suggest that these factors aren't just part of mydosomes, but they're actually active within them or being activated within these complexes. We next looked at their nanometer scale architecture, and for this we performed super resolution STAT microscopy, which gives us a resolution of up to 30 nanometer, which is absolutely incredible if you ask me, because this is still using visible light. And this showed us that MyD88 doesn't just form amorphous blobs in the cytosol, but it forms these defined barrel-shaped assemblies. And also do this with 2 colors and see where other factors land. And that showed us that the IREC kinases aren't just other core mydosome component, but actually within the topological core, whereas all the other factors and ubiquitin chains are on the outside cytosol-facing side. And lastly, since these are such highly complex assemblies, we next looked at the epistasis, so how does presence or absence of one factor affect the composition of these complexes. And we correlated this with the signaling output, for example here, the secretion of TNF-alpha. And what I want to point out here is that while these phenotypes are well known and characterized for many decades now, what we can do now is correlate them to the composition of the mydosomes, which showed that there's a high degree of programmability to these cytosolic signaling computers. And so with all this data in mind, I'd like to circle back to the question that I posed in the beginning. This is how we draw the toll-like receptor signaling pathway in the textbook, but is this how it really works? And my answer to this would be, from a cell biologically perspective, no, and that instead the entire TLR signaling pathway is executed from within the mydosomes. And with this, we ended up proposing a new model of toll-like receptor signaling, where activated TLRs seed protomydosomes that are released into the cytosol, and there they form these highly complex cytosolic signaling computers that contain all the factors that we need for the TLR signaling execution, such as the IRECs, such as the IKKs, such as the kinase TBK1, MAPKs, and later on also autophagy proteins. And this is the project that was funded by HFSP, and my fellowship just concluded, but I'm still working on this. And as with every science project, you maybe answer one question, you get 100 more. And what we're looking at now is, for instance, what regulates this release? Why do these complexes need to be released? What happens during infection? Can we try to build them in a bottom-up approach with synthetic biology? What are the activities in other cells, immune, non-immune cells? And taking a bigger picture question, why do we even need these highly complex signaling computers in immune signaling pathways when other pathways can do perfectly without them? With that, I'd like to thank my lab and all of our collaborators, the funders. Special thank you to HFSP for supporting me over the last 3 years and for having me here to present my work, and all of you for your attention. I'd be happy to take any questions.